Type 2 diabetes is a chronic condition that occurs in the body when it does not produce enough insulin or doesn't use insulin properly. Insulin is present and binds to its receptor, although the signaling downstream of the receptor is impaired. The important step of activating GLUT4 translocation to the cell membrane is also blocked. Glucose still in the bloodstream contributes to chronic hyperglycemia. The ligand is insulin, which comes from the pancreas behind the stomach. The ligand initiates the signaling process by binding to a specific receptor on the cell membrane, causing a conformational change in the receptor, which triggers a cascade of intracellular events. The primary receptor involved is the insulin receptor INSR, which sits on the surface of the muscle, liver, and fat cells, binding to the insulin as its ligand. A tyrosine kinase receptor is located on the cell membrane of the cells, like muscle and fat cells as well, binding to the hormone insulin as a signaling molecule. The protein kinase cascade affected is the insulin signaling pathway, where insulin binds to its receptor on the cell membrane, leading to a cascade of phosphorylation events, which results in glucose uptake into the cell. But in type 2 diabetes, this pathway becomes damaged due to the insulin resistance, which leads to reduced glucose intake and elevated sugar levels. Second messengers involved are 3,4,5-triphosphate, as known as PIP3, which activates downstream signaling molecules like protein kinase B that promotes glucose uptake. The normal response of the signal transduction pathway is a change in cellular behavior involving alterations in gene expression, protein synthesis, metabolism, cell growth, and differentiation. Insulin binds to its receptor on muscle and fat cells to trigger a cascade of signals leading to the translocation of glucose transports, also known as GLUT4, to the cell membrane, helping glucose uptake from the bloodstream. And the altered cellular processes are glucose uptake, glycogen synthesis, lipid metabolism, and protein synthesis. The signaling pathway is compromised due to insulin resistance in peripheral tissue and liver. This is caused by impaired receptor function, increased serine phosphorylation, chronic inflammation, oxidative stress, and lipid-induced toxicity. These disruptions contribute to impaired glucose uptake, increased hepatic glucose production, and abnormal lipid metabolism. The result of the disruption in the signal transduction pathway is usually called insulin resistance, or IR, shown in the picture above, and pancreatic B-cell dysfunction, causing cells to be less responsive to insulin, which causes high blood sugar levels because of impaired glucose uptake. This impacts the body because constant high blood sugar levels may damage the blood vessels, leading to complications with many organs. It can contribute to increased fat storage in the liver, and over time with type 2 diabetes, the body's attempts to make up for this insulin resistance by producing more insulin can exhaust the beta cell in the pancreas, which is what pancreatic B-cell dysfunction is.